In this video, I will be talking on the bioinformatics project on the annotation of genes involved with biosynthetic production of peptidoglycan within Merythermus ruber, involving supposed orthologous genes mRub 0981 and B1069, mRub 1162, and B065. Sorry, there's a typo there. mRub 1999 and B0084. To give a little bit of background on Merythermus ruber, it is a gram-negative non-motile bacteria, and when in a well-fed environment, it is in a rod-shaped form, and it is in a filamentous form when in a stressful environment. M. ruber has a growth range from about 35 to 70 degrees Celsius, with an optimal growth of 60 degrees Celsius. The bacteria has been discovered and isolated in thermal regions of Russia, France, and China, but were first isolated within the hot springs in a city of Russia. We chose to study this organism due to the lack of knowledge behind it. There are many bacteria in the world, like E. coli and Salmonella, that have had years of extensive research to explain hundreds of properties behind them. So making M. ruber our study organism will shed more light on what is unknown about it. And to do this, we'll actually be using those well-known organisms like E. coli. Well, we'll actually be using E. coli because it just makes a great model organisms. There's so much information already discovered about it that it makes a great model organism. This project revolves around the peptidoglycan production within Merythermus ruber. Peptidoglycan is a matrix found within the many layers that surround a bacterial cell. It plays an important role of allowing for molecules to pass through the membranes and prevents lysis of the cell wall due to osmotic pressure. When looking at the peptidoglycan layers, there is a difference in the amount a cell can have. This important aspect actually gave rise to the classification of a bacterial cell being either gram-negative or gram-positive. When you look at the gram-positive bacteria, you can see that the peptidoglycan layer is thicker than that of the gram-negative bacteria. This is the reason why someone who goes through the gram staining process have gram-positive cells that come out as purple and gram-negative cells that come out as pink. Gram-positive cells have a thicker peptidoglycan wall, which means that they're able to hold onto the stain more. They hold onto more stain than that of the gram-negative thin peptidoglycan layer. Here was a first step to our annotation. We were supposed to find genes involved with a specific metabolic pathway. I chose peptidoglycan biosynthesis, and I will later show the comparison between the M. ruber and the E. coli portions of the genes that I chose, and this region I'm circling around, these are the genes that I'll be focusing on. The first gene that I chose was the MVIN gene, which codes for lipid-2 flipase. Its function is to bring lipid 2 molecules that are essential for peptidoglycan production from the inner side of the membrane and expose it to the outer side. Whoops. Whoa. The next gene that I chose was the MERD-A gene. It codes for penicillin binding protein number 2, and it is essential for maintaining the rod shape of the E. coli cell and the M. ruber cell during peptidoglycan production. Next is PBPB, which codes for penicillin binding protein 3. Its function is to allow cell division of the bacterial cell during peptidoglycan biosynthesis. So I hypothesized that all the E. coli genes that I chose are actually orthologs to the M. ruber genes chosen, due to the background information that I picked up from the keg map. Here's the first step of our annotation. We took the two protein sequences between our sets of genes between the organisms and created a protein blast. And here you can see this is the result of the blast. We have a E value that is very low, very below the cutoff of 0.001, which is always good. And here we have similar, identical amino acids between the two. Here, and we also have these positives, which represent a similar characteristics between the two amino acids of the organisms. Here for our next set of genes, we still have that very low E value, and we have 29% identities, 43% positive. So there is evidence of structural and functional similarities between the two genes, which is good evidence. And here's our last set of genes, another very low E score, E value, and we still have similarities and identical amino acids. So far in the project, we had very a solid base of good evidence. Here was what I was speaking about earlier when I would be comparing the specific portion of the KEGG pathway between the M. ruber and the E. coli cells. As you can see, there are only 3 genes chosen, highlighted in these boxes, and E. coli actually uses those genes as well, giving stronger evidence that these genes could actually be orthologous to each other. Another piece of evidence that I found important was the PFAM alignments. The PFAM alignment consists of protein. It actually stands for protein families, and between these two genes, they both pull up the same protein family, the MVIN-like protein. And here we can see these alignments. We have sequences. We have the HMM consensus sequence, and the HMM. I mean, and the regular protein sequence, amino acid sequence for both M. ruber and E. coli. And I place X's above the conserved amino acids between the two species. They aren't linked up properly, but I tried my best to make it look like they're identical. We have the same here, except for this pair of genes, we actually have 2 domains. This panel A domain is the penicillin binding protein dimerization domain for M. ruber and E. coli, and for this domain we have the penicillin binding protein transpeptidase domain for M. ruber and E. coli. And as you can see, there are still a fair amount of X's showing identical amino acids. And here's the last protein family alignment. And all these X's being identical, excuse me, all these X's being identical shows that there are similar properties within both the proteins that these genes for E. coli and M. ruber produce. Another important piece of evidence was the transmembrane helice hydropathy charts. These red peaks show that the protein weaves in and out of the membrane, which would make sense because we're dealing with peptidoglycan production, which means that we're going to be dealing with proteins that are outside of near the membrane of the cell. And another thing with this set of genes specifically, the lipid 2 flipase, it would make sense because the protein brings lipid 2 molecules from the inside to the outside of the cell, so you would expect to see a lot of movement a lot of transmembrane movement. Here's the hydropathy chart for the next set of genes, we still have one peak showing that the protein would be localized around the membrane. And here's the last one, giving the same results. So basically, in conclusion, from using the different bioinformatics tools from the GenieAct website, I was able to predict that MRUB 0981 and B1069, MRUB 1162 and B0635, and MRUB 1999 and B0084 are orthologs. This was supported by the very low e-values we saw on the protein blast, and also through identical cellular location within the membrane, as we saw those transmembrane helice charts. We also use analytical steps like lipo-P and signal-P to give better understanding where the protein was in the cell, would be produced in the cell. And there are also trustful protein structures, like when we pulled up the cog numbers, all all orthologs pull up the same cog number. All orthologs also came up representing the same protein family, the P-fam, and all orthologs pulled up the same tiger fam number. There's also the same genes were expressed on the KEC pathway as I showed you in the in the previous slides, and also the P-fam alignments with the highly conserved amino acids. That was good evidence, and the hydropathy charts for the transmembrane helices dealing with peptidoglycan associated proteins. It would make sense that the proteins would be located in the membrane of the cell. And for a different approach to the experiment, Dr. Scott had asked us to find a way to disrupt the function of a gene, and I chose to disrupt the MRUB 0981 gene, the lipid 2 flipase, and we were supposed to use a specific site-directed mutagenesis step, and I chose to make a deletion of a highly conserved tyrosine at amino acid 346. And as you can see here, there is the amino acid sequence TAT for tyrosine, and what I chose to do is to make a deletion of that adenine so that the reading frame would shift and it would read a CTT. But the reason I chose tyrosine for it to be deleted was because of its affiliation with phosphorylation. Phosphorylation helps with signal transduction across the membrane of the cell, which plays a really good role with a protein that deals with peptidoglycan synthesis. And the reason I chose to change it to a CTT, which codes for leucine, was because of leucine's rare linkage to protein activity, making it a good substitute. And that is it.